Hello, sir. Good afternoon.
Okay.
Am I audible?
Audible?
Yes, sir.
One
One
second.
second.
So, very good morning, my dear friends. This is Dr. PADALA Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jagityal. Dr. P.Manjula Madam Garu, today's resource person. Madam is going to explain in the field of plant sciences, especially botany. Madam has clearly explained in previous class yesterday the complex tissue. In this complex tissue, we know that xylem and phloem, these are the two conductive tissue and complex tissues. Regarding of this complex tissues, Madam has clearly explained in previous class xylem. So xylem, it is a dead tissue. It contains different types of cells. That means tracheids, trachea, that means xylem vessels, xylem parenchyma, and xylem fibers. Madam has clearly explained yesterday. So, Madam, please welcome. Today, Madam is going to explain remaining complex tissue, that is phloem. Madam, please welcome. I have given sharing option. Please share your screen and start your session, Madam. Please welcome.
Okay, thank you, sir. Sir, is it visible screen?
Yeah, your screen is visible. Please click on slideshow, Madam.
Okay, sir.
Right.
Okay, good afternoon, my dear friends and students. So today's topic is phloem. Next topic, that is the phloem. Okay, so in complex tissues, so generally complex tissues means, so they are made up of more than two types of cells. So examples for complex tissues are xylem and phloem. So in yesterday's class, we discussed about the xylem and their elements. What are the components which are present in the xylem? So today's class, we will discuss about the phloem. Okay, so here these two complex tissues, that is xylem and phloem, commonly called as the vascular tissues. Okay, so the main function of these xylem and phloem is conduction of water and minerals and food materials. So the main function of xylem is, that is the conduction of water and minerals from the roots to the all parts of the plant, that is from the roots to aerial parts, total all parts of the plant. Whereas the phloem, it is the conduction of food materials. So the food materials which are mainly synthesized in the leaves. So they are here, they transfer these food materials from the leaves to all parts of the plant. Okay, so next that is the phloem. So it is a complex tissue and which perform a part of the vascular bundle. So vascular bundles means xylem and phloem. So it is also a part, phloem. So its components are the main components of phloem are sieve tubes, sieve cells, companion cells, phloem parenchyma, phloem fibers. So another name for this phloem fibers is phloem sclerenchyma. So all the components except phloem fibers are living. So whereas in xylem, so all are dead cells, only the xylem parenchyma is the living. But here all components are living except the phloem fibers here. Okay, so this is the main difference between xylem and phloem. So yet they form a part of the mechanical tissue system. So because these are living, but the main function of this phloem also, that is it gives mechanical support. So the phloem, it is chiefly responsible for the translocation of organic solutes, the elaborated food materials in solutions. Okay, so the elements of phloem which originates from the procambium of apical meristem. So this phloem which is derived from the apical meristem of procambium. Okay, so this xylem and phloem, they are developed, they are originated from the procambium or the vascular cambium. So here also based on the origin, this phloem, it is also classified into primary phloem and secondary phloem. So primary phloem means here the phloem which is originates from the procambium, it is called as the primary phloem, whereas the phloem which is originates from the vascular cambium, it is called as the secondary phloem. So similar in origin, that is xylem and phloem which is originates from the procambium, that is primary xylem and primary phloem which originates from the procambium, whereas the secondary xylem and secondary phloem which is derived from the vascular cambium. Okay, so another name for this phloem is the bast or leptome. Okay, so another name for the phloem is bast or leptome. So, these are used to refer the phloem. So bast, which refers to the phloem fibers, while the leptome, which refers to the soft walled parts of the phloem. So other than the phloem fibers like sieve cells, sieve elements, companion cells, and phloem parenchyma. Okay, so here generally phloem is present outside, xylem towards the inside and phloem towards outside. So it is called as the external phloem. So based on the position, here the different types like internal phloem, external phloem. So internal phloem means the phloem which is present internal to the xylem. It is called as the internal phloem. External phloem means the phloem which is present external to the xylem. So intraxillary phloem. So another term. So it is described internal phloem. Okay, intraxillary. So which is present inside. So interxillary. So with the phloem which is seen included. So in during the secondary growth, we can see this included phloem. So in between the secondary xylem, so patches of this secondary phloem is present. So it is called as the included phloem. So these are the different. So based on the position, so these terms are used: internal phloem, external, or intraxillary, or interxillary. Another name for this interxillary is the included. So which is present in between the secondary xylem. So we already discussed about this classification of phloem. So based on the development, here two types of phloem, that is the primary phloem and secondary phloem. So primary phloem means which is derived from the procambium. So during the primary growth, it is called as the primary phloem. And secondary phloem, which is formed from the vascular cambium, it is called as the secondary phloem. So generally this secondary xylem and secondary phloem, they are produced during the secondary growth. So next one is the components of phloem. So phloem, which is composed of several elements like sieve elements. So here the sieve cells and here sieve tubes and sieve cells, which comes under here sieve elements. Okay, companion cells, phloem parenchyma, and phloem fibers and sclerae. Another name is sclerites. Okay. So these are the components: sieve elements, companion cells, phloem parenchyma, and phloem fibers. Of which only the phloem fibers, these are the dead cells, and the remaining one, that is sieve elements, companion cells, and phloem parenchyma, these are the living cells. So this is the structure of the phloem. So it contains sieve elements and phloem parenchyma, companion cells. So here the sieve plate is also seen here. So first one is the sieve elements. So these are the living cells in the phloem, and so these sieve elements, they also contains protoplasm and no lignified secondary cell walls. And here the sieve elements are two types, that is sieve cells and sieve tubes. Okay. So first one is the sieve cells. So these are the narrow elongated cells without conspicuous sieve area. So in the sieve cells, that is sieve areas are not clear and they are taper at their ends and have a greatly inclined wall, so which overlap in the tissue. So these are the primitive one. Okay, sieve cells, these are the primitive one, and these are found among the members of lower vascular plants and gymnosperms. Okay, generally these sieve cells are seen in lower vascular plants and gymnosperms. Next one is the sieve tubes. So these are the advanced type of sieve elements, and generally these sieve tubes which are seen in angiosperms, and these are occur in angiosperms, and these are the longitudinal series of cells. So each cell is called the sieve tube member, okay, or sieve tube element. So because they are arranged in a series, so each sieve tube one by one, so they are arranged in a tube-like manner. So each member, it is called as a sieve tube member or sieve tube element. So here the sieve areas are seen, and these sieve areas are located on the end walls. So sieve areas are more specialized in the sieve tubes than the sieve cells. So sieve plate, that is a part of the cell wall which bearing the sieve area. Okay, and end walls of the sieve tube incline to transverse. So the sieve plate is the more specialized area found usually. So sieve plate, so it contains, so mainly these sieve plates mainly helps in the conduction, that is so these are nothing but that is they mainly helps in the transport of the food materials. So here, sieve elements, they are included in the sieve cells and sieve tubes. They include both sieve cells and sieve tubes. And from the ontogenic point of view, so this sieve cell resembles the tracheids. Okay. So in the xylem, that is first one, that is tracheids. So which are similar to these sieve cells, while the sieve tube resemble a vessel in the xylem. So sieve tubes, these are the long tube-like bodies formed from the row of cells which are arranged in longitudinal series. So where the end walls are perforated in a sieve-like manner. Okay, so here the sieve plate, it is nothing but the other perforated areas which are present on this. So through this, here the translocation of food materials will takes place. So the perforated end walls are called as the sieve plate. Okay, so sieve plates means they are the perforated areas through which, that is, cytoplasmic connections are established between the adjacent cells. That is the two cells which are connected with each other through these sieve plates. So sieve areas are comparable to the primary pit fields which are present in the primary wall with plasmodesmata connections. So when we observe in the cell wall, that is the connections between called as a plasmodesmata, like that. Here the sieve areas are also they also similar to that plasmodesmata of this primary wall. So the sieve areas are, however, more prominent and connecting the stands are widely are more conspicuous. So these sieve tubes mainly helps in the conduction of the food materials, translocation of food materials. So an insoluble substance called as a callose, which is impregnated into the cellulose or replaces the cellulose and forming a case around the each connecting strand, so which passes through the sieve area. So sometimes, so here the formation of callose will takes place. So due to formation of this callus, so it will not allow the translocation. So a sieve area in a surface view looks like a depression on the wall having a large number of dots. So when we observe this, so these are the different types of sieve areas or sieve plates. Okay. So on the wall having a large number of dots. So each dot represents the connecting cytoplasmic strands in the cross section and remains surrounded by a case of callose. So these are the, so these puppeting, so through which here the connection, that is the transport of the food materials will takes place. So see, the sieve plate or the perforated end wall is the primary wall of the two cells with middle lamella in between them. So the end walls may be obliquely or inclined or transverse. So end walls may be oblique or transverse. So the sieve plate, it is called as simple if it has only one sieve area, whereas the plate may be compound if there are several sieve areas arranged in scalariform, reticulate or other manners. So next, that is sieve cells. So we already discussed these sieve cells are comparable to the tracheids and are narrow elongated cells without conspicuous sieve area. So here in sieve cells, sieve areas are less or they are not present. Okay, so they usually have greatly inclined walls which overlap in the tissues, and there are more sieve areas at the ends. So sieve cells primitive than the sieve tubes, and the sieve tubes are enucleate at maturity. So they contain some nucleus during the development, but later at maturity the nucleus will be disappears, and there is disintegration of the nucleus with the maturity of the sieve elements. So the wall of the sieve element is primary and chiefly composed of cellulose. So the tubes cannot withstand the pressure from the adjoining cells, and they get crushed. So protoplasmic strands which pass through the pores of the sieve areas and strands remain surrounded by the callose. So with the differentiation of this tube, the amount of the callose increases and finally form something like a pad on the sieve plate. It is called as a callus pad. So due to its formation, the cell-to-cell communication is considerably cut. So due to this, as this formation of this callus will takes place, so due to which here that is the translocation will be stopped. So the callus pad is usually formed with the approach of resting or inactive season. So this is formed during inactive season and it disappears with the approach of active season. So this is called as a seasonal or dormancy callus. Okay. So in old functionalist sieve tube, the callus becomes permanent and it is called as a definitive callus or permanent callus. Okay, so involved, so sometimes, so here again callus, it may be seasonal or definitive callus. So seasonal means, so during the inactive season, so formation of this callus will take place, and during the active season, so this will be disappears. But here it will not, it will be permanent. It is called as the definitive callus or permanent callus. So due to formation of this callus, so it mainly the interrupt or the continuation will be stopped. That is translocation will not takes place. So next one is the companion cells. So companion cells remain associated with the sieve tubes of angiosperms, both ontogenically and physiologically. So they are small. So these companion cells, so the companion means they are always present along with these sieve tubes. Okay. So they are small elongated cells and having the dense cytoplasm. So it contains more amount of cytoplasm and prominent nuclei. Okay, they contain nucleus and starch grains are absent. Okay, so they occur along the lateral walls of the sieve tube. So these companion cells are always present along with these sieve tubes. A companion cell may be equal in length to the accompanying sieve tube. So sieve tube, the length of the sieve tube is equal to these companion cells. Okay, and the tube element or the mother cell may be divided transversely, forming a series of companion cells. So here the sieve tube element and companion cell which originate from the same mother cell. And the functional association is evident from the fact that the companion cells continue so long the sieve tube function. So it is present along with the sieve tube, and if the sieve that die when the sieve tubes are disorganized. So they are present along with sieve tubes as long as the sieve tubes are present. So if the sieve tubes are disorganized, or they will be disappear. So during that time, so these companion cells also disappear. So in transfer section, it appears as a small triangular or rectangular or polyhedral cell with a dense cytoplasm, protoplast. Okay. So when we observe in the transfer section, so these companion cells, they appear as a small triangular or rectangular in shaped structures with a dense cytoplasm, protoplast or cytoplasm. So generally these companion cells, these are absent in pteridophytes and gymnosperms. So here small parenchymatous cells remain associated with these sieve cells, which are called as the albuminous cells. So another term, that is the albuminous cells, which are present along with these sieve cells, and they die when the sieve cells become functionalless. Okay, so here companion cells are absent in pteridophytes and gymnosperms. So in that place, here the albuminous cells are present and they die when the sieve cells become functionalless. So thus the relation between the sieve cells and albuminous cells, it is similar to the existing between the sieve tube and companion cells, except that they have no common origin. Okay, so here the albuminous cells are comparable to the companion cells, but these albuminous cells and sieve cells, they do not originated from the same mother initial. So that is the difference between this albuminous cells and companion cells. So here this point is important. So generally albuminous cells are seen in pteridophytes and gymnosperms, whereas the companion cells are absent in pteridophytes and gymnosperms. Okay. So here albuminous cells are comparable to the companion cells because they are present along with the sieve cells in the pteridophytes and gymnosperms. But the origin is not similar. Okay. Because these albuminous cells and sieve cells, they are originated from different mother cells. Okay. But whereas in companion cells, so that is sieve elements and companion cells, they are originated from the same mother initial. That is the main point here. So thus the relation between the sieve cells and albuminous cells is similar to the ఈ పాయింట్ అర్థమైందా? సో ఇది ఇంపార్టెంట్ పాయింట్, దట్ ఈస్ albuminous cells are seen in the pteridophytes and gymnosperms, whereas in angiosperms in companion cells are present. Okay, so here albuminous cells are comparable to these companion cells, but ఇక్కడ albuminous cells, they are not originated from the same mother initial. Next, companion cells occur abundantly in the angiosperms, particularly in the monocotyledons. So they are absent in some primitive dicotyledons and also in the primary phloem of some angiosperms. So the wall between the sieve tube and companion cell is thin and provided with primary pit field. So along with these sieve elements, so these companion cells are present. So they are connected through these primary pit fields. So next element, that is the phloem parenchyma. Okay, so phloem parenchyma, these are the parenchymatous cells which are associated with the sieve elements, and these are the living cells with the cellulose walls and having the primary pit fields, and they are mainly concerned with the storage of organic food material. So the main function of this here in the phloem parenchyma, it is the storage of the food materials and also the tannins, crystals and other materials. So here the phloem parenchyma, it is also the living cell, and the cell wall is made up of cellulose and they have the primary pit fields. The main function of this phloem parenchyma is to storage of the food materials. In primary phloem, the parenchyma cells are somewhat elongated and occur with the sieve elements along with the long axis. But whereas in secondary phloem, this parenchyma is of two types. Okay, that is those cells which occur in vertical series, they are called as the phloem parenchyma, and others which are occurring in horizontal plane, they are known as the ray parenchyma or ray cells. So the cell wall is primarily composed with cellulose. Okay, parenchyma is absent in the phloem of monocotyledons. So phloem parenchyma which is absent in monocotyledons. So next one is the phloem sclerenchyma or phloem fibers. Okay, so among these phloem elements, that is the first one, that is sieve elements, companion cells, phloem parenchyma and phloem fibers, of which only these phloem fibers, these are the dead cells, and the remaining all are living cells. Okay, the phloem sclerenchyma, or another name for this phloem fibers is phloem sclerenchyma, because here these fibers which are made up of sclerenchyma, hence the name phloem sclerenchyma. So sclerenchymatous fibers which constitute a part of the phloem in a large number of seed plants. So they have rare in pteridophytes, and they occur both in primary as well as in the secondary phloem. So these phloem fibers, they are present in primary and secondary phloem, and these are the typical elongated cells having the interlocked ends, and here the lignified walls with simple pits. So here these are lignified and with simple pits. So the fibers of the primary phloem are essentially similar to those occurring in the cortex and secondary phloem. So there are considerable commercial importance as these fibers are abundantly used for the manufacture of ropes and cords. So sclerotic cells also present in the primary phloem, and they develop from the parenchyma with the age of the tissue, that is secondary sclerosis. So phloem fibers, these are the dead cells, and they are the sclerenchymatous fibers, and generally these are seen in the angiosperms and rarely in pteridophytes also, and they occur in primary and secondary phloem, and these are the typically elongated cells having the interlocked ends, and here mainly lignified walls with simple pits are seen. Okay, so these fibers of primary phloem are essentially similar to those occurring in the cortex. So the fibers which are present in the primary phloem, which are seen in the cortex and secondary phloem, similar to the cortex phloem or secondary phloem, and they are of considerable cost. So these phloem fibers commercially important, and they are mainly used in the manufacture of ropes and cords. So sclerotic cells, another one is the sclerotic cells. So they are present in the primary phloem. So they are developed from from the parenchyma. So this after sometime this parenchyma as during the it may after few days. So these parenchyma which are developed so sclerotic cells which are developed from the parenchyma due to secretions of the tissues. So they are called as a secondary sclerosis. So here, so this one is a phloem and so when we observe here the TS of the stem, helianthus stem, so this one is the epidermis, and here this is the secondary growth. So here the phloem fibers, phloem, and this one is the xylem, and central one is the pith. So we already discussed about this companion cells. So these are specialized parenchymatous cells found in slain that is associated with the sieve elements in origin, position, and also the function. And here the meristematic cells that gives rise to sieve elements. As such, a meristematic cell divides longitudinally, and one of them differentiate into sieve tube and other into the companion cells. So these are developed from the same mother initial. Okay, normally a single companion cell is found in a sieve tube in the primary phloem, but in certain plants such as in carrot, more than one cell have been reported. So, however, the solitary long companion cells are common in the primary phloem and in groups in secondary. So, generally in primary phloem, only one companion cell is present, whereas in secondary phloem, a group of companion cells are present. In a cross section, the companion cells appear as a round or polygonal or rectangular in shape. So, these are the living cells and have dense cytoplasm, prominent nucleus, and several vacuoles and other cytoplasmic organelles. So, starch grains are never found in the companion cells. So, these are the companion cells, and here the plasma desmata, sieve tubes, and parenchyma cells. So, phloem, it is one of the two types of transport tissues in the vascular plants. So the main function is conduction of food and organic nutrients from the leaves to the rest of the plant body. So, and composed of several types of cells. So based on the development, that is primary phloem and secondary phloem. So this primary phloem is derived from the procambium during the primary growth, whereas the secondary phloem, which is derived from the vascular cambium during the secondary growth. So this is about the phloem. Okay. So previous question, one of the previous question is the phloem of gymnosperms differs from the angiosperms in not having. Anyone? The phloem of gymnosperms differs from the angiosperms in not having sieve tubes, phloem parenchyma, companion cells, or phloem fibers. Answer? Anyone? Wrong answer. The phloem of gymnosperms differ from angiosperms in not having sieve tubes, phloem parenchyma, companion cells, and phloem fibers. So mainly, so these are here sieve cells are seen. Okay. So in gymnosperms, companion cells are absent. Okay. So in place of companion cells, albuminous cells are present. Okay. Here the answer is companion cells. Okay. When the xylem has a phloem on its both sides, the vascular bundle is known as. Okay. So that is on either side of the xylem, here the phloem is present. So this condition is called exarch, collateral, bicollateral, or endarch. Anyone? So on either side of the xylem, phloem is present. So it is called this condition exarch, collateral, bicollateral, or endarch. Anyone? When xylem has phloem on its both sides, the vascular bundle is known as good bicollateral. Parenchyma are found in xerophytes, zeophytes, lithophytes, hydrophytes. Parenchyma. Okay, parenchyma are found in xerophytes, hydrophytes, lithophytes, zeophytes. Good. Next. Intercalary meristems are found in lateral side of the stem, stem apex, root apex, base of the internode. Intercalary meristem are found at lateral sides of the stem apex, root apex, base of the internode. Intercalary. Intercalary meristems are found at lateral side of the stem. If the lateral side of the stem means lateral meristem, stem apex or root apex, apical meristems. Base of the internode, it is the intercalary meristem. Okay. Albuminous cells are found in phloem of monocots, phloem of dicots, phloem of gymnosperms. None of these. Albuminous cells. Okay, we already discussed this in today's class only. Albuminous cells, these are found in phloem of monocots, phloem of dicots, phloem of gymnosperms. Answer is answer. Anyone? Albuminous cells. Good. The phloem of gymnosperms. Okay. Only one person is active. Shaikh, your name is? Sheikh Kausar. Okay, good. Okay, so in next class we will continue with the special type of tissue. So these are also important special type of tissues. Okay, thank you. Sir,